silkworm egg. And inside the egg, the enzyme recognizes the familiar end sequences and cuts in it.
But it gets skinnier and skinnier.
As the globules go through the narrow duct, the liquid near the walls rubs against the surface, and it moves more slowly, while the liquid in the middle moves faster. So the layers slide past each other, which is known as shear. These shear forces stretch and deform the globules.
You also remove some water. So water goes out, you get a drop in pH, so it becomes a little bit more acidic as you go down this duct.
So the proteins are then being changed in two ways at once. Mechanically, they're being pulled and aligned, and chemically, that drop in pH means that there are now more positive hydronium ions in the solution. These bind to the negative parts of the proteins, neutralizing them. Now all the proteins don't repel each other as strongly, and so the N-terminal ends begin pairing, linking the proteins into long chains. These chains are pulled through a tiny narrowing called the drawdown taper, which leads to the spigot. Here the spider pulls the liquid through a small opening. This forces the liquid to speed up, and as it speeds up, it stretches. It's this final stretch that pulls neighboring spidroins tightly alongside one another. Now, depending on the sequence of amino acids, some sections fold into zigzag strands. These strands then interlock, and rows of hydrogen bonds stitch them into rigid sheet-like structures, which stack together into the nanocrystals that give the spider silk its strength. In other sections, the spidroins don't fold as neatly. That's how you get the amorphous regions, which give the silk its strength. Exactly how the proteins assemble inside the spinning duct is still being studied. No one fully understands it yet, and that makes recreating the process in a lab quite difficult. So scientists wondered, what if instead of starting from scratch, you used an animal that already has a spinner? This is exactly what researchers at Craig Biocraft Laboratories, a biotech company in Michigan, have been working on. What's more, they're doing it with an insect that humans have been farming for nearly 5,000 years. According to legend, a Chinese empress was relaxing in her garden when something fell into her teacup. She looked inside and saw a cocoon. The hot water loosened it, and when she lifted it out, it unraveled into a single shimmering thread, so long that it stretched across her entire garden. This was silk. Whether or not that story's true, people eventually learned to farm the insect that made the thread. Silkworms. They're the caterpillars of the domesticated silk moth. Silk quickly became one of the most valuable materials in the land. It dressed emperors and spurred trade connecting East Asia and Europe, so much so that it gave the trade route its name, the Silk Road. For nearly 2,000 years, China guarded the secret. But, of course, people were always trying to steal it. By the mid-6th century, the Byzantine Empire was in trouble. Plague had devastated the population, and on top of that, the purchase of silk was draining its gold. The problem was that Byzantines couldn't make silk. Most of it had to come through Persia, the very empire it was fighting. So Emperor Justinian made a secret deal with two monks. They would travel east, find the source of silk, and then smuggle it back. They returned with the secret hidden inside their hollow canes: silkworm eggs. This may be the first recorded case of industrial espionage in history. With that, Byzantium could finally make its own silk. Nowadays, we farm them by the millions.
One moth lays 500 eggs.
Okay.
So when you talk about like growing productions, right? Like, you know, my wife and I had a baby. These guys, they have 500 babies, and they do it every 30 days.
So the researchers at Craig wondered, what if we could make the silkworm spin spider silk instead?
When we create a transgenic silkworm, we're taking the genetics of a silkworm and the genetics of a spider, and we're putting those together. We're taking the best aspects of both. And that's what we're doing to create our fibers.
They start by gluing eggs onto a slide, which they line up under a microscope.
So this is the microinjection station. We're talking about eggs that are the size of the head of a pen. We're talking about needles that are half a micron in diameter.
This part is the I want to inject. So as you see, this is the tungsten needle, and this is the glass capillary. And first, we'll poke a hole with the tungsten needle. I will actually move the eggs to capillary and poke.
The fluid carrying the spider DNA is then injected into the part of the egg that will eventually become the silkworm. But at this point, the genes are still separate. They have to become part of the silkworm's own DNA. And luckily, nature already has a way of doing that. If you take an insect like a moth and look inside its genome, you'll see some stretches of DNA that move around. They jump from one place in the genome to another. These are called jumping genes, or transposons. One specific one is called piggyback, and it was first identified in a cabbage looper moth. Piggyback is a piece of DNA with two short sequences at its ends, and it works together with an enzyme that recognizes these ends, cuts the whole piece out, and inserts it somewhere else in the genome, kind of like a natural cut and paste system. What scientists realized is that if the enzyme only looks at the two ends, then you can keep those ends the same, but change what sits in the middle, and use that to implant the DNA of your choosing. So in the lab, the DNA in the middle is replaced with the spider silk gene. This, together with separate instructions for making the enzyme, is what's then injected into the silkworm egg. And inside the egg, the enzyme recognizes the familiar end sequences and cuts and inserts the spider silk gene into the silkworm's DNA. If everything works, it becomes a permanent part of the silkworm's genome. A silkworm where the edit worked looks almost exactly like one where it didn't.
The first approach we used was creating transgenics with the glowing cocoon. A green fluorescent protein is what's making these guys glow that green color under this particular wavelength with that particular light filter in front of it.
It feels magical. And there's a living thing inside of it.
Yep.
And it's creating the glowing effect.
Yeah.
But even if the DNA insertion is successful, it might not have ended up in the right place.
If I create the world's best recipe for chocolate chip cookies and I stick it in the pasta section, you're probably not going to make a lot of chocolate chip cookies, right? Because you're not going to see it.
Yeah.
Same thing for this, right? We want the recipe in the right section of the right cookbook.
That's why getting the spider silk gene into the silkworm isn't enough.
We really want the spider silk get incorporated into the silk gland, not any other tissue.
But Piggyback only looks for a 4-letter DNA sequence, TTAA.
There's so many TTAA in their genome, you couldn't really control where they were.
So it's like there are a lot of different places where they could park.
Yeah, that's true.
And that means that it won't spin pure spider silk.
If you had to estimate of the transgenic silk, like what percentage is spider DNA?
Like 6%, 10%.
That's insane to me.
So even with this limited gene transfer, the fibers still achieve much of the mechanical performance of pure spider silk.
Our average samples are usually about 60% of spider silk, right? Which, show me any other place in the world where you can get materials that perform at 60% the strength of spider silk that you can make cost effectively, right?
To push the 60% average even higher, the insertion needs to be more targeted.
The next step is to create pure, what are called knock-in, knock-out transgenics, where we will completely remove all aspects of the silkworm's native DNA for the silk and replace that completely with the spider silk protein.
And to do that, most of the leading labs in the field are exploring CRISPR-Cas9. This tool has 2 parts. The first is a guide, a short piece of genetic code designed to match one exact stretch of DNA. Unlike Piggyback, which recognizes a sequence just 4 DNA letters long, this guide matches about 20 letters, making it far more specific. The second is a cutting protein. When the guide finds its matching sequence, the protein cuts the DNA. Here, that spot is inside the silkworm's own gene for making silk. The cell immediately tries to repair the break, and scientists take advantage of this by also injecting donor DNA, carrying the spider silk gene. As the cell repairs the cut, it copies in that donor DNA, stitching the spider silk gene into this one precise location. This process is still in active development, and Craig is not the only group chasing the dream of spider silk. In Germany, AMS Silk is making proteins for fibers, coatings, powders, and hydrogels. And in Japan, Spiber is brewing protein fibers through fermentation. Brands like Goldwyn and The North Face have already used a version of these fibers in their clothing. And the military has been interested too. In 2016, the US Army funded Craig to produce ballistic shoot packs, panels layered with transgenic silk that are designed as potential body armor. And it's even being explored in medicine. For centuries, surgeons have stitched wounds together with silk. But now, a company called Neurotex is using spider silk to help repair damaged nerves. So if it can be produced cheaply, spider silk could find its way into clothing, armor, implants, even nerve repair. But to do that, we're gonna need a lot of it.
And this is our specialized spider silk. There you go.
This is the transgenic spider silk.
This is spider silk. This is transgenic spider silk.
This is like a bale. This is a hay bale of spider silk.
So last year we produced about half a ton of spider silk cocoon. And this is silk from that.
But what about our original question? Can you use transgenic spider silk to swing like Spider-Man?
So Derek, I've got you here in a climbing gym for a very good reason. And the reason is underneath this dirty dish towel. Unveil it, yeah, go.
All right, okay. This looks like very uninspiring.
So what's interesting is this is one continuous filament.
This is just one strand.
But each one of these strands is actually 10 individual silk filaments that are kind of connected.
All right, so you want me to hang from this?
I do want you to hang from it, yes. I actually want you to do more than just hang from it. I want you to swing.
This will be the first time ever anyone has ever swung from actual spider silk. Granted, this spider silk came from other organisms, but I don't know if it's gonna hold my weight, so let's see. In 3, 2, 1.
You really came close.
Don't tell anyone, but Spider-Man.
Okay, so we've seen it can hold your weight pretty reasonably. We could swing, but I do want to put it to the ultimate test and get you up there. How much do you trust Spider Silk?
Tensile strength doesn't lie.
All right, let's get you up there.
We used the title, Risking My Life to Settle a Physics Debate Too Early. Feels like it's gonna dig into my fingers.
This is crazy.
I'm gonna go.
Woo! Let's go.
You gotta see my finger, man.
Oh, damn.
Yeah, this is the problem with Spider Silk. It's thin, it's strong, and it just rips you open.
Now, if it isn't clear, we're not exactly web swinging experts, but we'd love to get the silk into the hands of someone who is.
Tom Holland is a challenge. Yeah, I'm calling out Tom Holland. I'm calling out all them guys. Andrew Garfield, Tobey Maguire. That's right. Come at me, bro.
See, spider silk is so strong and so thin that it actually cut Derek's skin. And that's actually a great way to explain the design flaw with modern razors. Thanks to Henson Shaving for sponsoring this video. See, facial hair resists more than you'd think, and many razor blades are engineered so thin that they bend under that resistance. This is called blade flex. Now, most razors actually build this flex in. They use springs, pivots, and flexible mounts that allow the blade to move. It's supposed to make the shave feel gentler, but it means that the cutting angle is constantly changing. When unsupported, the blade bends mid-stroke, and instead of slicing cleanly, it tugs at hairs, micro-cutting the skin. That's what causes irritation. The usual fixes are lubrication strips or extra blades, but they don't solve the real problem. The blade is still allowed to flex. We assume that razor bumps and irritation are inevitable, but that's shaped by bad design. But Henson is designed by aerospace machinists who control cutting forces with high precision, and their razors use a single, precisely machined blade. Instead of letting it move, Henson's design supports the cutting edge with tight machining tolerances, holding the blade securely so that the angle is consistent. Remove the flex, remove the irritation. Use code VERITASIUM for 100 free blades with any razor purchase. Just add both the razor and blades to your cart. Thanks to Henson Shaving, and as always, thanks for watching.